DNA replication is a fascinatingly complex process that combines the use of enzymes and many other different types of proteins. Replication was first proposed by Watson and Crick in the journal Nature, who said that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Replication of DNA copies the genetic materials, the nitrogen bases, or nucleotides, which process the information in DNA. Shagraff's rule states that adenine can only pair with thymine, and cytosine can only pair with guanine. Therefore, each of the new DNA strands created will have the appropriate complementary base pairs: adenine with thymine, cytosine with guanine. When the replication process begins, enzymes help the DNA unzip in several different places throughout the molecule. Then enzymes attach to nucleotides, which you get from your food, and they direct the nucleotides to the matching base pairs in the old strand of DNA. The new backbone forms from the phosphate group and the deoxyribose sugar, forming a phosphodiester bond, a very strong covalent bond. The nucleotides themselves form using a hydrogen bond, 3 for cytosine and guanine and 2 for adenine and thymine. This process is known as semi-conservative because one strand is old and the other strand is new. At the beginning of replication, enzymes help the DNA to unravel, creating a bubble. In the actual molecule of DNA, these bubbles appear hundreds of times because the molecule is extremely large, approximately 3 billion nucleotides long. Nucleotides, which are floating near the DNA molecule, attach to more enzymes, which attach to the nucleotides of the old strand of DNA. This process is enzymatic and you get the nucleotides from the food you eat, which is why malnutrition is very bad and nutrition is important. Then, another enzyme called DNA polymerase bonds the two strands together, and the phosphate groups and deoxyribose sugar bond to each other in a phosphodiester bond, which is a very powerful covalent bond in between each of the sugars and the phosphate groups. The nucleotides themselves bond together through hydrogen bonds, which help because molecules might need to read the DNA, and they would need to unzip part of the DNA and then zip it back up again very easily. If they had covalent bonds near them, they wouldn't be able to do that because covalent bonds are very hard to break and very hard to make. So hydrogen bonds, which are easier to break and easier to make, are the most optimal choice. The process of DNA replication is known as semi-conservative because the replication uses one new strand and one old strand, conserving the old one. Once the two new strands of DNA are formed, the DNA stays there in the nucleus until it needs to be used for the mitosis stage. This is usually happening during the synthesis stage of the cell cycle.
Hey, I'm Taylor and this is...
I'm Valeria.
Thank you for watching our videos. We hope you enjoyed them. Hope we see you again soon. Bye!